Well, hello, everyone. We're so excited that you're here for the Pacific Northwest Research Institute, or PNRI for short, Summer Undergraduate Research Internship Information Session. Before we begin, and I pass it off to our incredible investigators, I wanted to let you know that this session is being recorded. Your continued participation grants permission to PNRI to be recorded, photographed, or otherwise have images or voice recordings made, including but not limited to digital photographs, video, or digital moving images and or voice recordings for PNRI publication or promotional purposes in any medium, including but not limited to print media, newspaper, television, video, motion picture, website on the internet. Consent for use of your likeness or voice recordings is not a condition of employment, and this consent can be refused without any impact in the ability to fully participate in the SURI program. So thank you very much, and I'd like to pass it off to Dr. Rick McLaughlin.
Thanks, Lisa. Big thank you to everybody for showing up and for joining us for this session. It's really exciting to see people from all over the US, lots of folks in Seattle, from UW, I see some from Seattle U, from Edmonds Community College, Shoreline, Seattle Central, I think lots and lots of others. So thanks very much for coming out. We really appreciate you being here. So the goal of today is really to give you two things. One is a broad overview of what's happening at PNRI, the specifics of our internship program, what's happening in the context of our labs that you might become involved in, but also to kind of, in a sense, try to demystify a little bit this process that I think we all know can be quite intimidating of trying to land your first or your second, maybe, research internship in the context of a biological research program. So this session is meant to be informative, but also relaxed and informal. We'll have lots of time at the end for you all to ask whatever questions you might have for us about the science, about the Institute, about your experience in trying to apply for these sessions, and anything else that might be of relevance in general. I'll add that feel free to go ahead and type any questions into the chat that you might have. We'll save those questions for the end, and those questions can, again, be about the science or about anything else that comes up in the presentation. Okay, so we are Pacific Northwest Research Institute. PNRI is the oldest nonprofit research institute in Seattle. We are made up at this point of 5 different labs that are really united in our interest in thinking about genetics and genomics. And as you'll see from the research talks, the topics that are encompassed in PNRI's interests are very broad, very diverse, everything from very applied and clinical human genetics all the way to thinking about contagious cancers in clams. And that's about as broad as I think you can get in the context of a research institute. So for me, it's a really exciting place to be because of that you get exposed to all these different topics at different scales and different styles. So we're all here today to talk about SURI, which is the Summer Undergraduate Research Internship. The reason that we host SURI is because we are extremely vested and dedicated to providing research experiences and access to those research experiences to as many young people as possible. And in particular, we're interested in expanding that access to students who have been or currently identify with groups who have been historically excluded from the sciences, but also students who may not have access to these sorts of research experiences at their university. And in particular, we're really trying to get more community college and two-year students involved in these internships. And as a result, you'll see that while our internship very much mimics and replicates what happens in other institutes, we have some nice pieces that are really built to try to accommodate students who, for example, really need to be paid over the context of the summer and can't really afford to not have a job and do these sorts of unpaid internships. So some technical details. The SURI is a 9-week program. It's a research intensive program. And that means that the main component of the program is being placed into one of the labs, one of the 5 labs that you'll hear from in a few moments, where you'll have a mentor who's your day-to-day mentor that you talk to about, what you had for breakfast, but also the experiments that you're thinking about, what you're thinking about in the context of your career, the culture of the lab. And you'll have sort of a PI mentor, which are the heads of the lab as well. but you're really paired into this somewhat close relationship with at least two people in the context of your lab. But that is the keystone of the internship, and you'll have a project that's assigned to you that will really be yours, that you'll be an intellectual owner of, that you'll work on in the context of the lab. But in addition to that research piece, we're also extremely interested in exposing our undergraduate interns to the diversity and variety of careers that maybe you could participate in if you get an undergraduate degree in one of the sciences. So while we have all gone on to be lab leads who are very interested and invested in doing biological science research, there's just a multitude of careers out there of people who have had backgrounds that are quite similar to ours, but go on to do different things like, undergraduate teachers, patent lawyers, project managers at Microsoft, and a lot of the skills that you acquire in the lab or in your biological sciences-based degree are highly applicable to lots of different degrees and lots of different careers. So one of the things we call it the career lunch. The undergraduate interns meet with a different speaker each week, and that person is someone from the PNRI community, or more likely the broader community, who has a science background, but now does something that is not typically a lab leader. You get to sit down, talk to them, learn about their background, learn about why they do, what their job is like. What does it really mean to be a scientist in all of these different career environments? So in addition to that, we have a research intern symposium that happens at the end of the summer. Sounds a little scary, but it's actually a really cool place where you get to present your science that you've worked on so hard over the span of the summer, in a very supportive and encouraging environment, and we get to see all the really cool things that you've done. There's a lot of social activities as well. So in addition to the really core research activities, we try to create this environment where you're going to have fun, you're going to socialize, you're going to be able to think prospectively about what you might do in the next step of your career by interacting with lots of different people who have gone through lots of different forms of science education. Okay, so a couple more details. You have to have completed at least 1 year of undergraduate study. That could be at a community college, a 2-year university, or a 4-year university, but you must be at least finishing your freshman year to be eligible for this internship. These are paid internships, so we think it's really important to pay the interns in order to really allow this opportunity to be accessible to as many people as possible. So the total payment is a $5,600 payment. That's one nice thing about it is that you sort of get a check on the day you show up, and that can really help you get started in the summer without having to wait a full month for your check. The internship is 9 weeks long. That's a little flexible in terms of your university schedule, when that starts and when that ends, but we do require that you dedicate at least 9 weeks to the internship. And on average, interns work about 30 hours per week. We also have some transportation options where we can give you a free Orca card to facilitate your getting to and from the internship. Okay, so what is this internship all about? What do we hope that our interns gain from this experience of 2.5 months in the context of the lab? The first is, and I think this is something that I always say to my lab, we want you to learn to read, write, speak, and do science. So science, I think, and especially biological science, can be really intimidating. And there's a lot of jargon, there's a lot of technicalities. And an unfortunate side of it is that sometimes you have to master that a bit to really be able to delve into the details and the concepts and the fun part of thinking about these really important problems. So you will absolutely gain an intense amount of experience in that part of the science. And that's going to help you a lot in the context of careers in your classes as well. You'll be exposed to all sorts of different scientists from different careers, from different backgrounds who have studied different fields of research. And you'll see that there's not really just one kind of scientist. All of us on this call have very different backgrounds in terms of what we studied, what we're interested in, how we think about problems, where we grew up. You'll leave with a newfound confidence, I think, in your ability to do this sort of laboratory work. And I know Amy Dudley likes to say that one of the best things that we can see as an outcome from this internship is that somebody leaves here and they're ready to be a really, really good undergraduate research technician, either in the context of their university or as a technician after they graduate. So we really try to give you those skills to have confidence and the abilities needed to do that sort of a job. We think you'll gain all sorts of new friendships and interactions, both in the context of the scientists at PNRI, but also your cohort of interns. And finally, it's super important to have fun. So nobody around here sort of takes life too seriously, and we're all, relaxed and excited and engaged in our work in a way that we're very excited to share with folks from all different backgrounds and experiences and interests in the context of the science. Okay, so in the next part of the talk, I'm going to turn the microphone over to my fellow scientists. We're going to go through each of the PIs. We'll spend 3 to 5 minutes telling you a little bit about the very broad questions that their lab is interested in thinking about, and also some of the very specific projects that you as an intern might be able to work on in the context of their lab. So Claudia, I believe you're up first. And for the PIs, I'll control the slide. So if you just want to say next slide, I'll go ahead and click it over to the next one. All right, Claudia.
Yes, okay. Hi, thank you, Rick, for the awesome introduction of the SURI program. So my lab studies human genome variation in genetic diseases. We study rare diseases and a lot of different types. Many of the rare diseases we study affect children. So we have a lot of pediatric cohorts here. And we study structure variants in the human genome. I don't think I can show any. Okay, so I cannot point to the slides, but the first one is what we study. Most of us in the lab study structure variants, which means that we are studying the DNA alterations, like copy number variants, can be deletions and duplications and type of gains, translocations to other chromosomes, insertions, and inversions. So we characterize those type of alterations using different types of sequencing technologies. We have a couple of them in the lab. We also have arrays and digital Doppler PCR. So the students who come to my lab, they usually get a wet bench experience, but also some bioinformatic experience analysis. And why we do that? Because the different types of alterations in the DNA, they can cause functional consequences and potentially functional consequences that will have a phenotypic consequence for the patient. So we are either trying to find what alterations are those, or we are trying to understand how those alterations are actually impacting the phenotype. So for instance, one type of structure variation that we study a lot are duplications and deletions. And they usually affect, when they are causing phenotypes, they are usually affecting those are sensitive gene, or a gene that cannot be deleted or a gene cannot be duplicated. Those are the type of genes we are studying. And we also study how those alterations are formed. So there are a few mechanisms that are known to cause these alterations. One of them is called non-allelic homozygous recombination. The other one is non-homozygous joining, but there are others that we study as well. And we want to understand what are the instances and the contexts where those alterations occur in the DNA. They can be in the mitosis, they can be in the meiosis, and that's all important for the disease and also for counseling the families. So that's what the what and why and the how of the lab. Rick, next, yes. The potential projects for summer undergrad researchers would be solving the molecular puzzle of unsolved rare diseases. There are many diseases that people don't know the molecular cause of those diseases. And we look specifically in the DNA to try to find clues. For that, then we have the wet bench part and also the bioinformatic part where we have to establish the genomic pipelines. And that's one of the projects that the student will be involved. The other part of the lab is studying alterations that we found. And for that, we have here an example of the Robinow syndrome, where we found each one of the genes shown in this cartoon is we found alterations that cause the disease and affect a specific pathway called Wnt pathway. and cause a disease that is a skeletal dysplasia called Robinow syndrome. We found alterations in each one of those genes in patients, and we are trying to understand why those alterations are causing disease. So we're doing functional studies on them. So that's another part of the lab that will have projects for people to work. I think that's it. Thank you, Rick.
Thanks, Claudia. Amy?
Yeah, hi, my name's Amy Dudley, and I'm the head of the Dudley Lab. In my lab, we use the awesome power of yeast genetics, and that is in fact a real term that people, serious scientists use. And the reason people call it the awesome power of yeast genetics is because yeast is, and when I say yeast, we're talking about baker's yeast, so the kind that you all got familiar with when you were baking, making bread and in the pandemic. So baker's yeast. So you might be surprised to know that there are actually hundreds, even thousands of labs across the world that use baker's yeast as a model system. And the reason for that is that the fundamental processes that happen inside a yeast cell also happen inside a human cell. And so much that we know about human biology was actually learned by studies in yeast. Several Nobel Prizes have been awarded for studies that were done in yeast. So yeast has similar biological processes, similar cellular processes to those that happen in humans and human cells, including the processes that when they go wrong, they cause disease. And so one of those sets of processes that our lab focuses on a lot is the process of various processes in metabolism. So this is the production and utilization of various nutrients inside your cell. And again, in yeast and human cells, these processes are so similar, and the genes that encode the proteins that carry out, for example, the enzymatic reactions are often so similar that one can actually replace the yeast ortholog, so the yeast version of that enzyme, with the human version of that enzyme inside the yeast cell. So what we do is we delete the yeast gene. Rick, if you want to kind of hover over, we'll test you to see how well we've drawn these diagrams. So we delete the yeast gene from the yeast genome, and then we take the human version of that same gene, and we actually clone it into the yeast genome, the human gene into the yeast genome. And so if those processes are really similar enough, and we've engineered the cells correctly, then the human enzyme will be able to function inside the yeast cell. And when that happens, we have what's called a complementation assay. So for example, if the enzyme is responsible for producing a particular amino acid, let's say the amino acid serine, when it has the yeast gene, it's able to grow at 100% when we don't give the cells serine. But when we delete the gene, now it's non-functional and the cells can't grow. And when we put the human gene back, the human version of that enzyme back in, now we're able to restore growth under that specific condition. And so this is really powerful. And there's a group of really dedicated people in my lab, including our lab manager, Julie, who is really good at art. And so Julie has drawn these representations of all of us, including our superhero persona as she sees it. Next slide, Rick. So if you join my lab, in addition to getting your own Marvel superhero persona done by Julie, you will hopefully learn and get access to a number of different opportunities. So many of the projects that we're focusing on in the lab, and the types of projects that you would work on if you were an undergraduate intern in our lab, deal with our studies of using these very powerful yeast assays to try and understand, not even try to understand, to actually understand which of the many variants in the human genome cause disease and which of them are completely harmless, and everything in between. And so one of the things that you would do in our group would be to try to set up these kinds of assays for a particular human gene, a particular human disease, and then make variants in that human gene that correspond to variants that are actually seen in real people, sometimes in real patients, sometimes just in random people in the population across the world who have had their genome sequenced, making those particular mutations and then testing them using these yeast assays. In addition to knowing a lot and learning a lot about biology, human biology, yeast biology, kind of the biology of metabolism and how these things work, you'll learn a lot about molecular biology and high throughput techniques. So the field of synthetic biology is generally kind of this type of taking an artificially a piece of DNA that's been made in a factory, actually in San Francisco, and then putting it into a yeast cell. That's generally called genome engineering or synthetic biology, and we do all that all day, every day. So you'll learn about a lot about that. Our lab uses a lot of robots, so we have several different robotic systems that we use in order to generate and assay these yeast cells in high throughput. We use a lot of automated image analysis and different kinds of DNA sequencing. So Oxford nanopore long read sequencing is our favorite in the lab, and other groups at PNRI use it and other things. We use automated image analysis, computer programming, so lots of different kinds of computer programming. And these are all techniques that we don't expect you to know. So we are going to teach you everything that you might have had a little experience maybe with molecular biology, maybe you've run a gel, maybe you've programmed in Python before, maybe you haven't done any of those things, but we're going to teach you all of that. So that's kind of one project. Another really cool thing that has been enabled especially by virtue of some of the artificial intelligence applications into biology, particularly with respect to the applications in protein structure, is that we have at least predicted, sometimes real, but sometimes predicted protein structures for all of the yeast and human genes that we study. And so one of the other set of projects in the lab is actually focusing in on using protein structure and the analysis of protein structure to help us in our design of some of these assays, and also some of our interpretations of some of the results that we get from these assays. And so even though I am somebody who takes everything very seriously, I hope to see you all, or many of you, at the internship, and thanks for joining the webinar.
Thanks, Amy. All right, so my lab, the McLaughlin lab, is a picture of my group here. We are, I would say, at heart evolutionary biologists, but we're really interested in understanding parts of the genome that are not genes. So we're all familiar with this concept of a gene that makes RNA, that makes some protein, and that protein does something in the context of the organism. You know, maybe it encodes eye color or kidney physiology, something like this. But we actually study another portion of the gene, another portion of the genome that we call transposons, or transposable elements. So just like a gene, these are pieces of DNA that make RNA, that make protein. But the big difference now is that these parts of our genome, they don't really do anything for our body. They don't really do anything to support our physiology. All they do is they go and they make another copy of themselves. So these are selfish elements, and they exist because they can copy themselves inside of our genome. And this might sound a little wacky, a little zombie-like, but we actually know that these replicating virus-like things that are inside of our genome have created way more than half of the DNA content of our genome. So while protein-coding genes like this constitute about 2% of our genome, we think maybe as much as 60 or 70% of our genome has been created by these replicating sequences called transposable elements. So we are really interested in thinking about not just what these sequences are doing. They also do one other interesting thing, which is that sometimes they accidentally bind other RNAs in our cells, and they make new copies of genes in our cells. So all the time, our genome is changing. Our genome is not this static picture that you see in a textbook, but everybody's genome is different. And even in the lifetime of a single individual, our genomes are changing. And part of that change is actually the creation of new genes in our genome. So you don't just have a given set of genes over your lifetime. You might get new ones over the lifetime of species on the context of millions of years. We know that, for example, humans have about 7,500 additional copies of some of these genes. Most of them don't do anything, but within there, there's a lot of interesting biology happening. So our lab is really interested in thinking about a couple aspects of this. How are these transposable elements, these selfish sequences, changing in human genomes? How do different human genomes differ in their composition of these selfish virus-like sequences? And we actually have a really amazing resource now in that there are more and more human genomes all the time that we can leverage to look at these sequences. So one project you might do in our lab as an undergraduate is look at the diversity of some of these transposable element sequences across very diverse human genomes and try to understand something about how they've changed in the past and how they might be active now, both in healthy individuals, but also how they might be activating in inappropriate ways to drive diseases like autoimmunity or cancer or other diseases. So like Amy mentioned, there's a lot of computational biology in our lab, but this is something that we're very excited to teach people. All you really need is an interest in thinking about how to use these computational-based techniques to think about biology. But pretty much every project in the lab has two sides of that single coin, where we use the computational biology and we use the genetics and the evolution to pose interesting questions and generate hypotheses that might not be really obvious just based upon wet lab molecular biology techniques. So a lot of the times you'll be going back and forth between genomes, tissue culture, cell culture, biochemistry in the context of the lab. Another project we're really interested in thinking about is how these new copies of genes that I showed you, called retrocopies, might be impacting our ability to maintain health when we're infected with a virus, or alternatively, might impact our ability to fight off those pathogens and increase our susceptibility to certain infections. So I think it's really interesting to think about why certain individuals are more susceptible or resistant to a given infectious disease. And one reason is that their genomes are different in ways that result in that difference in susceptibility and resistance. And we think these retrocopies are one way that might be driven in the context of humans. So these are just two examples of projects, but most of the time in the lab, again, our projects are really rooted both in using computational methods and wet lab experimental methods to tackle these different questions about genome evolution. And Lisa?
Okay, so I'm Lisa Stubbs, the leader of the Stubbs Lab. And our goal is to study the functions of genes that are associated with human neurological disorders. And our focus is really on disorders such as autism and things that usually come along with autism in its various spectrum. There are people who are just a little bit afflicted by autism and people who are very seriously. They often have other problems like high anxiety, very high susceptibility to stress in their lives and so forth. And we want to understand how those genes that are associated with those diseases actually function. What do they do to your brain? What do they do to the individual's brains? And what are the mechanisms by which they do that? So we're really interested in particular in the genes that are called gene regulatory proteins like transcription factors and chromatin modifiers that control the activities of other genes in a hierarchy. So they will actually turn those other genes on and off. And very often the genes that are the master regulators that I'm talking about, plus the genes that they regulate, are all individually can be associated with neurological disorders. They act together and they can all contribute to the disease. So there's lots of work out there to identify human mutations. We're also a little bit involved in those studies, and many other individuals are doing those kinds of human mutation studies. But once a gene is identified, you need to find out what it does, how it works. And so what we do is we use mouse models to try to understand how those genes work. So once we have the mouse models, the first thing we look at is how does it affect brain development? So that image is a thin slice of a mouse brain. And the colors are markers that are expressed in the mouse brain that we can apply to the thin slice and image it under the microscope. And so this is actually called the hippocampus. It's a really important region for learning and memory, and also other kinds of behaviors, and all sorts of important functions, and got a very interesting structure, which is why most people like to focus on that region. It's very, very orderly structure. And when people have mutations that affect that structure, you can actually see it under the microscope. You can see the cells are different. They're in different locations. There are different numbers of them. They express different proteins and so forth. And so that's one of the first things we do when we get our animals that have a mutation. The second thing we do is we test their behavior. And so the middle image is with the mice running around. We videotape our mice as they behave under certain circumstances. And then my staff scientist, Chris Seward, is a really whiz-bang computational biologist who has developed some AI-based tools, or adapted some AI-based tools that allow us to monitor the mice as they move around in the cages and to quantify their behavior. But we do a lot of the quantification also just by hand. People will analyze the mice and look at the mice, and sometimes you can pick out things with your eyes that AI just can't do quite yet. The third thing we do is we look at the brains and look at their gene expression patterns and the molecular biology. So we want to understand, all right, we've knocked out this gene, we have a problem in the structure of the brain, the mice behave strangely. What's the basis of that on the molecular level? So we do all these three things. And depending on where we are in the project, we will focus on one or the other. And the lab does all of these things. We do molecular biology, we do behavior, and we do a molecular pathology to try to understand what these genes do and how they function. So next slide. So there are two big projects in the lab, and I won't go into detail on all of this text here. It's just kind of there for your reference. We are interested in, as I said, the kind of master regulators, the genes that control other genes. And one of those genes that we focused on is a gene called OTS2. And OTS2 is a gene that makes several proteins, and they're all involved in regulating chromatin. So they turn other genes on and off, but the different proteins, the short, and we call them isoforms, the short and the long isoforms have different functions. So OTS2 is associated with autism, ADHD, and a lot of other neurological disorders. Depending on where the mutations are in the gene, you get different effects. Some people have epilepsy, some people have things like bipolar disorder, and some people just have the kind of standard kit of autism and ADHD. A lot of individuals have learning difficulties as well. It's doing that because it's a gene that controls very early basic steps of neuron development in the brain, and where the neurons go, how they connect with each other, and so forth. So it's really got a fundamental purpose. It's a very large gene, and as I said, it has the two isoforms, and the longer isoform is something that we've studied a lot, and others have too, and we've recently published a paper on its function. We kind of are beginning to understand how it works. But the shorter one is completely unstudied. And so this project that you'd been involved in would be characterizing this very new mouse mutant that we have that nobody's ever developed before. It's a completely unique resource that we need to characterize so we can understand what this shorter isoform does. And we know that the short and the long isoform work together, and you can't get the complete picture of how this gene causes neurological disorders and what it does in the brain until you understand both parts. It's been really difficult to get something to target the short isoform, but we have it now. So that would be something that a person would be involved in. The other project that we have in the lab is, why are certain individuals more susceptible to stress than other people? And how is that linked to downstream problems with coping and metabolic disorders and so forth? So whole body health. And we've identified some genes that are involved in stress response. And so one of the things we're doing is subjecting mice to particular situations that they find a little bit stressful, and trying to understand exactly which parts of the brain are most important to their stress response. as I said at the top, all the projects are team efforts. What you would do in particular under these, you definitely would just be embedded in the lab. You would learn all of these different things. You'd be exposed to all these different techniques. And depending on what your strengths and weaknesses and interests are, your background and so forth, you probably slot into a smaller part of one of these projects, but you'd be working as part of the team. And so all of the people in my group do pretty much everything I described. We do molecular pathology, we do behavior, we do animal handling, we do just, trying to understand how the animals function. And we do molecular biology. We do a lot of molecular biology to try to understand what the mechanisms are. So those things are all going on in the lab, and what you would be involved in would be a little bit dependent on what's going on at the time in the lab and what your interests are and what your background is. So that's it.
Thanks, Lisa. Michael?
All right. Hi, everybody. I'm Michael Metzger. So I'm the last one to talk about lab. So I am going to tell you a little bit about transmissible cancers in clams, and that's the main thing we work on in our lab. So I should start off by saying cancer is not normally transmissible. So cancer cells do not normally jump from one person to another or from one animal to another. We know of things that are infectious diseases, viruses like HPV that can jump from person to person and cause cancer. But when they do that, the cancer cells will always grow and develop from each new person, or in animals, cancers normally grow from each new animal. What we're studying here is an unusual phenomenon where the cancer cells themselves actually jump from one animal to the next and spread through the environment. And so we've found this in clams, soft-shell clams on the East Coast, a number of other bivalve species, including mussels, and most recently, this cockle species that's found in Puget Sound. We've found cancers in a number of different locations in Puget Sound. And so we're trying to understand this in a number of different ways. I mean, one way is just trying to understand how these things work and how they exist and affect the environment and the ecosystem and aquaculture. And then the other side is trying to understand these as models to understand how cancer can work and how cancer can interact with hosts, the animals that, or humans that have the cancer, to try to see if we could use understanding of this to help human health. And so what we really do in order to identify one of these transmissible cancers, there's a little sort of schematic here that in normal animals, when you look at all of the cells within a normal individual, whether it's a person or a clam, essentially there's differences between one individual to another, but for the most part, all the cells in one organism are the same. They all have what we call the same genotype. But when we see these animals with cancer, this transmissible cancer, we find that the genotypes, the genetic sequences in the host, the clam, do not match those of the cancer at all. And that when we look at the genes and the sequences in the cancers, all the different cancers from multiple animals, from even multiple locations, will have the same genotype. They'll be nearly identical. And that shows that you can that what's happening is that these cancer cells are actually jumping from one animal to the next. So we have a wide variety of work in the lab, going from some actual field collection to computational work. Let's see, we've got, we have a little example of a computational phylogenetic tree showing the evolution of these transmissible cancers and how they're diversifying into two different groups in soft-shell clams. We have a picture of what the cells look like under the microscope. And then we even have some experiments in the lab where we keep clams in the lab. We experimentally inoculate them with cancer and are trying to understand how the disease progresses in different populations. And then we even collect samples from a bunch of collaborators sort of, and collect them locally in Puget Sound, as well as from a number of different collaborators really all over the world. And there's one really interesting example we found of a cancer in mussels, where it seems that the same transmissible cancer cell that evolved in one mussel has spread not just between populations or different individuals, but all around the world. There's this one lineage where it's the same one found in Europe and in South America. And even other collaborators have found the similarly lineage in Asia as well. So we've got a lot of different projects that are potential projects that, like we said, you don't have to know how to do these things. We can teach you all how to do it, and that you'll be sort of embedded with some of these other researchers who are working on these. To look at the genetic diversity in cockle cancers in Puget Sound, as well as other places in the world, using environmental DNA techniques to track cancer spread in the wild. So this is actually sampling water from a number of different locations, and then we can extract DNA from that and identify the sequences specific to the transmissible cancers in that water, and use that to identify where the cancers are and how they're spreading through the environment. And then just using other methods, looking at the spread of the soft shell clam cancer and the cancer in mussels. And then we're also trying to get these cells to grow in the lab. So that's looking at tissue culture and ways to try to grow them, which is going to be a really useful tool for us in the future, as well as genomic analysis of the cockle and mussel cancers in particular, because we're just getting a whole bunch of sequence data, and we're finding out an enormous amount about how these cancers are able to mutate and how they change over time. Finding things like unique mutational signatures that are in some cancers, sort of pretty unexpected amounts of instability in these genomes, and actually able to sort of date these cancers. At least in the soft shell clams, we were able to date it, to say it is approximately 400 years old. That's the first one we've analyzed. And so that is telling you that sort of the first clam that had this cancer was about 400 years ago. And they've been spreading as a cancer cell through the environment since then. And really, that we know that for the clam one, but we don't know anything really about the mussel and cockle cancers. We're just starting that. So there's a huge amount of questions and a whole bunch of open questions that we need to work on. So there's a lot of variety available there.
All right, thanks very much, Michael. Okay, so hopefully this part of the information session has given you a nice overview of PNRI as an institute, the goals and composition of the SURI program, and a little bit about some of the specific projects and specific lab topics that could be encompassed in the context of your internship with PNRI. So in the last 10 minutes or so, we want to switch gears a little bit and talk not just about applying to a PNRI SURI, which we certainly encourage each of you to do, but also some tips and guidelines on how to think about your application materials when you are applying to a number of different undergraduate internship programs. So start with the PNRI specific application. So you can access this website either by going to this link or scanning this QR code. In order to apply to PNRI SURI, you do need to submit your internship documents directly through our website. And the required documents are first a resume, and I'll talk very briefly in just a second about what we hope to see in the context of that resume. And second, this one-page statement or cover letter that describes how participating in this internship will enhance your education and career goals, and specifically which labs you're most interested in. And in a moment, Amy will tell you a little bit more about how to think about that one-page statement or cover letter, both in the context of your PNRI application, but also other applications you'll be submitting. Okay, so when you go to apply to these programs, there's typically some subset of these three application pieces that you'll be required to submit. The resume, the cover letter, or the one-page statement, or the personal statement. It's called a number of different things. Sometimes you'll be given particular questions as prompts. Sometimes it'll be a little bit more open-ended. And third, a letter of reference. So you do not have to submit a letter of reference to the PNRI application program, but if y'all have questions about that at the end, happy to answer questions about the letters of reference as well. So the first piece in your application packet is really the resume. And this is obviously the classic document that you'll use in every job application you submit, probably for the rest of your life. So what we want to see at this point is a concise and minimal version of why you think that you're well suited and why you think you're going to be a really good candidate for the positions that we're posting. So some of these qualifications include your education. Where are you in school right now? What year are you in school? what work experience have you had? And this isn't just, what scientific research work experience you have, but what has been your lifetime of work experience? I think that can be a really relevant piece of information for thinking about how you'll be able to engage in these sorts of research experiences. Third, what classes have you taken that might be relevant for this internship? That's usually the hard science classes, but might be other classes that you can imagine could be relevant for any particular experience. In addition, any outreach, community service, or additional activities that you have participated in so far in your career and in your education. And finally, just anything else that you feel like shows a unique aspect of you or something that would help us understand who you are as a candidate. Okay, so at this point, I'll turn the mic back over to Amy, who will talk to you a little bit more about the one-page statement or the cover letter. Amy?
Yeah, thanks, Rick. Can you advance the slide?
Ooh, too far. There we go.
Great. So, so one of the reasons why we felt it was really important to talk about the cover letter is because we frequently see applications where, unfortunately, the applicant didn't understand that the cover letter is actually, in some ways, the most important part of the application. And this is because, this is really, it's your place to shine. It's your place to, a place where you can boost your application to the top of that pile. And one of the reasons for that, there are a lot of reasons for that over the course of your career, but at this point in your career, everybody, all of the applicants that we see, you guys all have a limited amount of scientific experience, and that's by definition, because that's what we're looking for as applicants at your career stage. And so everyone really has about the same level of experience, and everybody pretty much has, the same coursework. And you can't control, kind of what your GPA is at this point, at the point where you're, applying. But this is something that you can actually control, right? You have the freedom, you have a blank slate to make the best case for why you should actually get this position. And then the final thing is that, this is a helpful tool for the committee, right? So we are going to look at 1, maybe 200 applications. And we're looking through a lot of them. And then we have the ones that are, kind of at the top of that pile. And then we have to distinguish amongst those. And so reminding us in the cover letter that you have this or that experience, which you think, helps you, or this or that about yourself. If it's right there in the cover letter, then when we're talking about you and reviewing the applications, that reminds us. So even if you think, oh, I told them that already in the resume, tell us again, tell us the important stuff again in the cover letter. Okay, next slide, Rick. Yeah, and so this is here in the recording. We're going to go through this with an example. So I'd like to go through kind of with 2 examples. One that is kind of an example of a weak cover letter, and one that's an example of an exceptionally strong cover letter. And we're going to go through these various aspects of kind of what makes something a strong cover letter versus a weak cover letter. So if we go to the next slide, Rick. All right, so we receive applications like this, okay? And unfortunately for poor Greg here, they didn't realize that, the things that I've just told you, which is the cover letter is really your chance to shine, and the cover letter is really important, okay? So what are some of the things that Greg did here? So Greg didn't tell us anything about themselves. They just said, "Dear hiring manager, I would like to apply for the internship, and, I can start in June. So of course, I'm the only application you're going to get for this. And there's no reason for me to kind of make myself stand out. And here it is." Okay. So that's really unfortunate for Greg, because he's unlikely to make it to the top of that pile. And in fact, in the case of this internship, the cover letter is absolutely required, and Greg is actually not going to make it to this, our top of our pile. Okay. So let's go to the next cover letter. Okay, So this is the next cover letter, and this is actually a cover letter written by a University of Washington, in this case, undergraduate. So this cover letter has really most of the things that we are looking for in that A+, amazing, standout, top of the pile cover letter. Okay, so the first thing is that they made an attempt to address it to an actual person. So they showed that they understood something about kind of who they're applying to. So for example, if I it would not be a mistake, even if you don't think that Michael Metzger is the only person reading this application, you're interested in applying to Michael's lab. So dear Dr. Metzger would be a perfectly reasonable way. And even if I'm reading it, that doesn't matter. It's shown that you kind of understand kind of who you're talking to. At the very least, when you're writing these applications, if you don't know who to address them to, dear members of the review committee might be a good one to talk about if you're talking about like an internship program. Okay. So the next thing that you want to do in a strong cover letter is tell us right up front kind of what position you're applying for, and in this case, for example, what lab you're applying to. So in this case, and I specifically have not chosen an application that is applying to the SURI program at PNRI. So this is an application for a student who is not applying for an internship. They were applying for a position in undergraduate research in somebody's lab, and they're not applying to PNRI. So what are they applying for? They're applying for the research opportunity. It's never a bad idea to say where you heard about their research opportunity. So that's not required, but that's a little helpful for us actually to know kind of, is everybody hearing about it through this undergraduate research database, or is everybody hearing about it on Handshake? That's kind of helpful, but that's not super. So now super important. So tell us something about yourself. So Miles here has told us that they're a freshman with junior standing at the University of Washington. What is your major? As Rick was saying, kind of where are you in your undergraduate career, right? So I'm, Miles is currently a freshman with junior standing, interested in becoming a physician scientist, and here's their major or planned major. Great stuff to know. The part that really separates kind of the B+ applications from the A+ applications, so rising to that level of kind of a B+ application is the applicant has at least looked at our website and knows a little something about them. So instead of I'm interested in science, I am interested in your lab because I know that you work on virology, for example, might be kind of the B+. In this case, and you could read this, this student actually had a really thoughtful discussion of what about this person's lab interested them, and in addition, and this is really the A+ paper, A+ application, find one paper from that lab that really speaks to you in some way. Now, in this case, the paper that spoke to this person was this very kind of, technical, I'll be honest with you, I don't know Dr. Octavius's work, but this is not their top paper. This is a paper that was a really kind of technical small paper for this lab, but it really spoke to this student. And the reason it spoke to this student is one that they talked about. And so I'm not looking for somebody who has found the most prestigious paper from my lab that's talked about in my New York Times, article about me in the New York Times. I'm looking for kind of what speaks to you. And so this letter does a really nice job of that. Then the next thing is there something else that would be helpful for us to know, right? So in the case of, and this might be a little, maybe not so much for the SURI program, but what kind of courses have you taken, right? So summarize kind of the kinds of courses. And biology 327 doesn't mean anything to us or anybody else, right? But I've taken these, so Miles here has taken courses in biology, chemistry, calculus, and statistics. And so we have some sense of what their background is. Is there something about the that we might want to know about you. So for example, you know, kind of when can you start in the lab might be important. In this particular case, apparently the job ad had something about, you know, being able to accurately pipette. Ours aren't going to say that, but let's say that there was something that was listed in the job ad, and gee, you really don't have experience, or you think, you know, maybe that you want to let them know that this isn't, you don't quite have this experience. And so, you know, I thought that what this student did was actually really helpful, which is, you know, although I've had limited opportunities to practice pipetting because of COVID-19, I have strong attention to detail, a basic knowledge of molecular biology, and a drive to work hard. And that was really great. And, you know, again, this is, you know, something that a student actually wrote. So that was very thoughtful. And then finally, and this is important also for the SURI program as well, you know, kind of what do you hope to get out of this experience? So these are meant to be educational experiences, just like working in an undergraduate research lab would be an educational experience. And so what do you hope to learn, and how might this help advance your career trajectory, educational and career trajectory as you see it? And then thank them for their you know, for reviewing your application, and then, you know, sincerely, your name. So that's this kind of an example of, you know, kind of an A+ kind of letter that's going to get to the top of our pile.
Thanks a lot, Amy. And again, this will be recorded, and I think we're also going to send out the slides to the attendees. So the tips and pointers here will be highly accessible to y'all. So at this time, we want to take a moment to thank you all for coming, and thank everyone at PNRI for organizing this. And we have time for questions now, so I encourage you to raise your hand. I'll call on you. You can unmute, maybe even.
Well, I have one from the chat. Lewis has asked, approximately how many interns would be working in each lab?
So we, our cohort size is a little bit variable year to year. I think we're probably looking at somewhere between 1 to 2 interns per lab, but that's in a cohort of 5 to 10 interns in the whole institute.
Great. All right, Iris has asked, so for this internship, who should we write our cover letter to?
You could write it to members of the review committee. You could write it to the scientists at PNRI. Or if you're targeting your interest to just one lab or 2 labs, I think it's fine to address your cover letter to those lab leaders, the PIs. So if you're interested in Amy's lab exclusively, you might address your cover letter to Dr. Amy Dudley.
Thank you.
And I think the main point there on addressing that, Amy, feel free to chime in, is to show that you be not completely generic, but to show that you have an understanding of who you'll be working with, who the composition of the review committee might be, and who the people at the institute are, something that's not completely form lettered.
All right, I don't see any hands raised, but we do have some questions in the chat. So Lauren has asked, how many total interns are you looking to select? I think you mentioned between 5 and 10. Is that right, Rick?
Yeah, that's correct.
Great. Okay. And I apologize in advance. I believe it's pronounced Daya. What does your specialty of interest mean in the application form?
Lisa, do you know what she's referring to?
Now, Iris, or I'm sorry, Daya, I think it's area of interest, and the scientists are probably better suited to answer this question because I'm just an HR lady. But Rick, why don't you go ahead and field this one?
Okay, sure. Sorry, I wasn't sure. But in the submission page, one of the fields is your specialty of interest. Yes, I think that's a slightly odd wording, but you could put, molecular biology or human genetics, or sort of, if you're a computational biologist, or if you're a computer scientist, or if you're a bioengineer, your major would be a fine thing to put in there, but your very broad area of interest.
Great.
Sorry for the confusion there.
All right. And Marshall has asked, can we find your scientific papers on the PNRI website free of charge, or will we need a subscription?
You can find lists of our papers on our websites. You can also find them on PubMed. And because the vast majority of our research is funded by NIH, almost all of our publications will be posted as open access prints on PubMed. If you, for some reason, can't find one, feel free to email us and we're happy to send you a copy of it as well.
Wonderful. All right. Aram has asked, would we be considered for other labs if we selected just one lab in the cover letter?
So you do need to declare which labs you're interested in. Because of the number of applications we get, we really allocate each intern's application to the labs that they select as being of particular interest to them. So you're welcome to say that you're interested in more than one lab and state exactly which labs. But I would state which labs you're interested in discreetly and not say something like, I'm interested in all of them, even if you are. It really helps us for you to prioritize your interests a little bit as we go through, a lot of applications.
Great. Adriana has asked, what is the best preparation you recommend for this internship?
Amy, do you want to take that one?
Yeah, well, so again, we are really, we're going to show you everything that you need to know. So, so you don't need to, cram, your biochemistry 101 textbook, and you don't need to start, teaching yourself Python. But prior to, if you are selected for a certain lab, one of the things that we will do in advance is to reach out to you. And some people, for example, feel more comfortable if they've read a couple of relevant review papers on the subject area, right? So these are papers that might kind of generally go over kind of important things to know about retrotransposons or mouse genetics. Sometimes you might be, some people would like to read a couple of papers from the lab. And so, so at the point where you're, where you have been chosen as an intern and you have selected a lab, you will be interacting with your direct mentor and most likely the head of the lab. And they will get you all set up in advance so that you know what to expect when you arrive on day one. But just be assured that we're going to teach you everything that you need to know. So, yeah.
Wonderful. Thank you, Amy. We have an anonymous question, and that is it advantageous to decide which lab you're most interested in?
It is essential. So you don't necessarily have to say one. You have one page of the cover letter to convey your interest in a particular lab. So there is a bit of a trade-off. If you choose multiple labs, that you might be able to write a little bit less. But if you put 2 labs, maybe even 3 labs, we do put your application into the pile for each of those PIs. So I don't think there's a particular advantage, except that you might be allotted more space in your cover letter to talk about your interest there.
Okay. Maya and another participant asked this question. Are Running Start students able to do this program as well? So if they have completed the equivalent to 1 year of college as part of the Running Start program, are they also eligible?
I think we're happy for you to apply. We typically do look at those students who maybe have actually completed a full year of on-site university study. And that's really just for the sheer reason of at some point we have to make cutoffs somewhere, and those students that are already in college have fewer opportunities and fewer chances to have these internships. But very happy to receive your application and take a look at it as well.
Okay. I'm not sure if I'm pronouncing this correctly. Iran has asked, will we be working with grad or medical students in addition to professional scientists and fellow interns?
Yes, absolutely. So the composition of the PNRI is a lot of professional scientists who are research technicians, staff scientists, but we also have graduate students from the University of Washington, from the molecular cellular biology graduate program, from genome sciences, from bioengineering, a number of other programs. We also have undergraduates from UW who work in the lab, and I don't think we currently have any medical students, but it might be, you might interact with someone who is heading off to medical school next year or doing a post-bac fellowship on their way to medical school. In the context of their career lunches that are also a part of the internship, you will almost certainly interact with all of those categories of people.
Wonderful. Nathan has asked a question for Dr. Dudley. He indicates that he might have missed this, but for the slide where it showed the yeast enzyme and the human enzyme rate being 100% and 90%, how come, Dr. Dudley?
Yeah. So although sometimes, so in these enzyme replacement assays, the enzymes usually match very, very well, but they're not usually perfect. So there are subtle, sometimes major differences. And so if something works at 90%, that's actually really good. And we can work with that in our complementation assays. So there are differences between being a human and a yeast cell, as much as I hate to admit it. But and so sometimes those enzymes are a little bit more optimized to work better in the human or the yeast cell. And that's where those quantitative differences can come into play.
Thank you. Naomi has asked, is there anything specific you're looking for and anything I can do to help bring attention to my application?
Amy, do you want to take that as a follow-up to your cover letter?
I mean, so we are looking for your interest in doing science in the particular labs that you've identified. We are interested in supporting your goals for your educational experience kind of as you move on into the sciences or using your science background in some other career. And so we're looking for you to tell us how this opportunity, as you understand it, might help advance your educational and career goals. The other thing that I wanted to mention, and this isn't, I don't know if this is the exact right place to kind of point it out, but while I remember it, Rick, you talked about how in some applications for some programs, there will be a letter of reference that will be important. And at some point when you're applying for other jobs, other research positions, people will ask for letters of reference. One of the real positive effects of being in an internship program like this is you will get to know scientists like the PIs and like your personal day-to-day mentors. And those people will be able to write letters of reference for you for future job applications. And that is something that is not a hassle. It's not something that is a real imposition on us. That's something that we expect to do. It's something that we enjoy doing. We have gotten to a certain stage in our careers because people have have supported us in these kinds of ways. And we're happy to use our privilege and our influence to help advance your career. And so we'd like to hear kind of what interests you and excites you about the position, and how do you envision this helping you in your educational and career?
Wonderful. Thank you. Angela is asking, will there be an interview for the applicants you've chosen?
Yes. So after the initial review of the applications, we'll reach out to a subset of applicants for one-on-one or one-on-two interviews between the PIs and perhaps other members of the lab with those applicants chosen for an interview.
And it won't be scary, I promise. Nathan is asking, I have a question for Dr. Stubbs. Has there ever been a neurological disorder related to, I'm not sure if I'm pronouncing it correctly, Gilal, G-I-L-A-L, cell malfunction?
Glial cell, yeah.
Glial, okay, thank you.
Yeah, yes, in fact, many. The neurons in the glia usually interact with each other very closely, and they have really important interdigitated functions. And without glia, the proper glia and proper activity of glia, the neurons don't survive. So even in cases where the neurons end up being the thing that looks abnormal, it could be a result of a malfunctioning glial cell. So a lot of the genes that we study are expressed at the really early stages of neural stem cell development, where they can go to become a glial cell or a neuron. And so the genes end up being important to both cell types. And so many disorders affect both neurons and glia, and there are lots of cases also where it's just the glial cells that are abnormal, and the neurons by themselves would be completely normal. It's just they don't get the support they need from the glial cells, so they end up being affected too.
All right. Thank you very much. And our last question is from Lauren. Do you prioritize applications from students of a specific university, for example, University of Washington, since you mentioned having grad students from there?
The answer is no, very broadly. We like to have folks from a diversity of different institutions of different sizes and styles from all over the United States. I will mention that we are applying for funding from National Science Foundation that would provide funding for interns, specifically earmarked for interns who come from community college and two-year programs. So we are very interested in providing this opportunity, like I mentioned, to students who might not have these kinds of experiences at their home institution as at least a part of the cohort. But overall, we're really interested in having students from universities all over the US.
Thank you. And actually, we've got one more question. This is a late breaking question for Dr. Metzger from Nathan. Do you happen to know where the origin of the Puget Sound's cancer variety came from? He indicated he couldn't tell from the map.
That is correct. We can't tell either. So yes, there's a lot of questions about that. We don't know yet.
Okay. Well, those are all the questions. Rick, I'll hand it back to you.
All right, so thanks very much, everyone. A big shout out to Lisa and Gina for helping organize this. It's been really successful, and a huge thank you to all the PIs, and a very special thank you to all the candidates who came out. We hope this was informative. If you have additional questions, we have the communication via email, a couple addresses that you're welcome to reach out to with anything you might have about your application or the program or any technicalities or details. We're very happy to help you with that. We highly encourage you all to apply to the program, and we hope to see your applications in the application pool. Thanks very much.
Thank you.